I think we are at a turning point with stem cells, and if we manage to have this maturation, it will completely change the game. Usually, if you want to develop some drugs, you need some animal testing. But for cardiac tissue, it always fails. I would say often fail. So you need to develop other models. And one thing that is really interesting is you can use human stem cells from patients, for example. But in that case, you need to make the differentiation of the stem cells into cardiac tissue and after to make the maturation.
Contractility is one of the biomarkers which is related to viability and maturity of the heart tissues. And it is different for healthy and disease states of the cardiomyocytes. And also many drugs aim to modify this property. So by studying this property in vitro on heart-on-a-chip technology, we can assess the efficacy of drugs.
The human cell-based models are far more realistic than those in animals because the cells are derived from humans. So we really have the same cell signaling system, we have the receptors and the ion channels that are the same for humans.
The aim of the project was to obtain the maturation of cardiac tissue, I would say. And to obtain this maturation, you need to couple chemical stimulation, electrical stimulation, and mechanical stimulation. And it's very, very difficult to have both or to couple all this stimulation in the same device.
So the project's goal is to reach from this young stage to a more developed and more adult-like phenotype. And at that point, it's a very good model to replace primary cells. And the most important, that it has human origin.
This is the insert system that we developed. So it's basically made up of mainly two pillars that hold this little electrospun mesh. Mesh is composed of nanofibers. And then we attach this little mesh into the system. And then we seed cells into that mesh. And then at some point, by stimulating the cells, they start beating, and then these little pillars here start moving. And then that's how we can analyze how the cells are behaving.
The cells were seeded on these scaffolds, and then we applied electrical and mechanical stimulation. And without electrical and mechanical stimulation, these cells are very difficult to mature. So that's why we found all possible directions for maturation and combined all of them in this one device.
The application of this model has a vast application capabilities because these heart-on-a-chip models can be used for new drug development, for biomarker development, for understanding the mechanisms of diseases, and also for then we can try to find new targets for the drugs.
So the results of the EMAPS-Cardio will provide a platform which is more relevant to human studies and human drug development. And with that, hopefully we can shorten down the drug development process, make it more cost-effective. Hopefully there will be less clinical failures on the cardiac side effects.
Our system is now optimized for measuring micromotion in the valves, and which is good for cardiomyocytes. But we can also think of contractility of other tissues like lungs or muscles. For that, we would need to modify magnification factor, which is possible because the design is configurable. So we could imagine to use this system just for imaging of organoids or some microorganisms in multiple plates. Yeah. So we're looking for exciting applications.